Transformation is a process in which a cell is altered so that it can acquire and express genetic material obtained from the surrounding medium or environment. The most common use for transformation is the introduction of recombinant plasmid DNA into bacteria. Bacterial strains can either be naturally transformed or can be made competent by artificial means. Transfection, on the other hand, is the introduction of foreign DNA into the nucleus of eukaryotic cells by non-viral methods. The most common gene transfer methods are chemical, lipid, and physical methods. There are many different methods to transform cells. However, not every method can be applied to both prokaryotic and eukaryotic cells. To transfect eukaryotic cells, electroporation and sonoporation are commonly used. Electroporation is a physical transfection method that consists in the application of electric pulses to create temporal pores in the cell membrane. It can be used in bacteria and eukaryotic cells, and it's the most efficient technique. Sonoporation can also be used as a transfection mechanism, and it is an ultrasound-mediated cell membrane permeabilization, or induction of membrane pores using ultrasound. The pulses of ultrasounds are delivered by a machine, and there can be of selected intensity and duration, depending on the sample. There are several things that must be considered in order to select a bacterial transformation method. First of all, it's important to consider the method through which the cells have become competent. For example, when making chemically competent cells, the use of calcium chloride is required, and high salt concentrations in the medium will damage the electroporation equipment and the cells, so heat shock transformation will be the best option. Also, the transformation efficiency required is something that should be considered, because electroporation can provide higher efficiency than the chemical transformation. Another thing that should be considered is the cell membrane and the wall composition, because of some cell components can be resistant to certain transformation methods. The ideal method for cells that present resistance to other transformation and transfection methods is electroporation, because it facilitates the pore formation in the wall by the electric pulse. Another important factor to consider before choosing a transformation method is the size of the plasmid that will be introduced into the bacteria. Electroporation is a good method for high molecular weight vector transformation. Vectors with a molecular weight of 50 kilobases could be transformed by this method. Finally, it's also important to take into consideration the capacity of the cells to resist the damage caused by the transformation method in order to ensure that the cells will be alive after the transformation method. Apart from the most common transformation methods like heat shock and electroporation, there are other methods that can be used, like bioballistics, that is a mechanic method that works like a gene cannon, in which DNA is wrapped in gold or tungsten particles, then it's disposed in a machine that uses compressed air to introduce the pellet in the cells. Plate transformation, which is a method that consists in preparing a culture of receiver bacteria and incubating it at 37 degrees and 200 rpm overnight. Afterwards, they must be translated to a new medium and incubated at 37 degrees and 200 rpm for 2 or 3 hours. 1 milliliter of the culture must be centrifugated and resuspended in 200 microliters of LB medium, mixed with 10 nanograms of plasmid, and finally speculated in cold plated with calcium. Colony to lung transformation, which is a method used when a plasmid is in a bacteria and it's necessary to introduce it to another one. This method avoids the destruction of the plasmid that is necessary to transform competent bacteria. This process needs to take the donor bacteria and dissolve it in water and ethanol, add calcium chloride and mix with the receiving bacteria, and then transform. When making calcium chloride transformation will be the best option. Also, it's something that should be the cell membrane and components. The transformation. The ideal method of other transformation is it facilitates the electric pulse. Another important factor is molecular weight vector. Finally, it's caused by the transformation methods. There are also mechanic method that works like a tin can wrapped in gold or to use air to introduce that consists in translated to a new medium translated to a 7 degrees and 200 rpm 3 hours. Suspended in 200 microliters of medium. Coloring method used in a bacteria and it's necessary to introduce method avoids the transform. Afterwards, they are to a new medium and incubated at 1 milliliter must be centrifugated and resuspended in 200 lb medium. Coloring to lawn transformation, which is a method used to introduce it to a plasmid that is necessary for an ethanol at calcium.
After centrifuge, the supernatant was eliminated. Then 10 milliliters of calcium chloride at 0.1 molar was added in order to resuspend the pellet, and then it was incubated in ice during 10 minutes. The sample was centrifuged at 4,000 revolutions per minute. Then the supernatant was eliminated in order to resuspend the pellet with 2 microliters of a solution containing 0.1 molar calcium chloride and 15% of glycerol. Then the sample was incubated in ice for 10 minutes. For electrocompetent cells, after centrifuged, the supernatant was eliminated and then 10 milliliters of a cold glycerol at 10% was added in order to resuspend the pellet. The sample was incubated for 10 minutes in ice and then it has to be centrifuged at 4,000 revolutions per minute. After the second centrifuge, the supernatant was eliminated and the pellet was resuspended with 2 microliters of cold glycerol at 10%. It is important to resuspend and disaggregate the entire pellet formed in the bottom of the tube and also the mass that is in the walls of the tube. For both electrocompetent and chemocompetent cells, different aliquots were made in order to preserve the cells to use them in some other experiments. 50 nanograms of a plasmid were added to the chemocompetent cells. Then they were incubated in ice for 10 minutes, and then they were introduced in a water bath at 42 Celsius degrees during 45 seconds in order to produce a thermal shock. 300 microliters of sock medium were added and then the sample was incubated at 37 Celsius degrees during 30 minutes with shaking. A sample of 40 microliters of electrocompetent bacteria and plasmid were added in an electroporator cell. The electroporator was set at 1500 volts in order to give the pulse to the cells. 300 microliters of soak media were added and the sample was incubated at 37 Celsius degrees during 30 minutes with shaking. Finally, the cells were cultured in LV plates and incubated. Incubated for 12 hours, we observed the colony form unities, and in order to obtain the efficiency of both cells, we count the CFUs, obtaining 188 CFUs in the electrocompetent cells and 134 CFUs for the chemocompetent cells. The efficiency values obtained talk about low efficiency in both electrocompetent and chemocompetent. However, analyzing the UFC formed in the plates, we can say that the electrocompetent cells were more efficient than the chemocompetent cells. This can be because of the transformation method. Also, this can be possible because of the sterilization methods or the use of sterilized areas was incorrect. And then this can be also because of a bad use or technique with the culture of the cells with the bacteriological spatula.
The method of transformation that will be selected must have in consideration the steps, regions, and purpose of the cloning. Heat shock transformation and electroporation methods differ mainly in the methodologies used for the preparation of competent cells, as well as the method used to induce temporary pores in the membrane, which could be heat of an electric pulse. To prepare chemically competent cells, it is necessary to incubate the cells with a calcium chloride solution or another salts, while the preparation of electrocompetent cells only requires a wash and an incubation in a glycerol solution. For chemical transformation, it is important to respect the time and the temperatures for the heat shock step, and for the electroporation, it is necessary to ensure that the sample is free of salts and place it at the bottom of the electroporation curvette.